In this lab, you are going to be finding the different stages of cell division in onion root tips. You are going to be looking for cells that are in interphase, prophase, metaphase, anaphase, and telophase. The first thing you are going to do is take a slide, make sure that your microscope is set to low power. That will be the red-tipped one. Then what you'll do is put the slide in and look for thin, small roots. Sometimes they're green, sometimes they have a red tint to them. Once you're focused under low power, then move to high power. Once you're in low power, then go to medium power, then go to high power. Remember, under high power, you only are to use the fine adjustment focus knob. Do not use the large one. Use the small knob on the back. That's the only thing that you focus on. If you can't find it, go back to low power, try again. Now, once you're under high power, you need to find cells that are going through the particular stages. Now, you don't have to find them in any order. You can find them in any stage that you want, whatever stage you want first. So if you wanted to find, for example, metaphase first, you could do that. What you're going to do is take and move the pointer and the slide so that the black tip pointer is over one of the cells that you have located. Then you're going to call me over and you're going to say, Mr. Curtis, I think that this is, and then tell me you think it's metaphase or anaphase or whatever. And then you're also going to say whether you think it is early in the stage, middle of the stage, or late, because these go slowly through each of the stages. Then I am going to put a mark. On the sheet, I will say it's either right on, yes, you're absolutely right, you're sort of close, or not really. If you get a mark in the sort of close or not really, you get to try it again to get full points for this lab. Again, if you have any questions, call me over and I'll be happy to give you some assistance.